And we'll continue up. Oh, the poor little kid, his what salivary gland is getting all inflamed in this little dude here?
The parotid?
That's the parotid. The parotid is the one that's kind of right here in the side of your cheek, kind of pouring in behind your molars there. What are the ones that are right underneath your jaw here?
The lingual?
Those are the submandibular. And you also have that right under your tongue, the sublingual. So, and now we're also in the digestive tract, but this time it's an actual photomicrograph. So what do we call, let me get my little annotator again. What do we call this layer here that I have highlighted? It's getting folded into this thing here. What is this?
Submucosa.
That's all submucosa, which is made out of
Dense irregular CT.
exactly. So, and this is where you have a lot of times glands are down in there. There's all sorts of blood vessels and things like that. Nerve plexuses, that's where the submucosal plexus of Meissner is found. We're going to look at this actual structure in more detail later today. This would be an example of one of the plicae circularis, kind of the large circular folds. What do we call these finger-like projections where the mucosa comes up and down into these little fingers?
Villi.
Those are the villi, exactly. And we'll see. So by looking at this, what part of the digestive tract are we in? Does anybody have an idea? What part actually has the villi, the ones that have little finger-like projections? This is more of a spoiler. We're gonna look at this in much more detail today.
The small intestine? Small intestine.
Yeah, this is the small intestine. The small intestine is the one where the mucosa is thrown into the little finger-like villi. We could probably say even more, this is more likely the jejunum because the fingers are not so dense. When you get, not the jejunum, the ileum, when you get to the jejunum, there's so many of them, it's really hard to get a nice clean cut like this where you don't just have a whole bunch of like sliced off fingers in the view. So, excuse me, let's clear that. Continue rolling, rolling, rolling. And what organ are we looking at here?
Salivary gland.
Yeah, this is total classic salivary gland. And, and if you were using your, you're thinking about it, the S was actually the serous cells, the M were the mucus cells, demi was a serous demilune, SD is like a salivary duct. So the lighter staining cells are making a what? These are they're making the mucus, they're the mucus cells. And again, we're gonna see similar looking cells just along the lining in the rest of the digestive tract where they're goblet cells. Goblet cells are basically single-celled mucus glands and they're just scattered all over the place. And fine, then we go, why name the bone-like material that teeth are made out of? And that was the dentin or dentine, they're both similar. And then this part, I put this in, again, it's not worth points, but I think it's worth starting to really remember or remind you guys, the final is coming up in 3 weeks and the final goes back to the beginning. So these would all be fair game, like on, the final exam, again, outermost layer of bones, the periosteum, that should still kind of be ringing bells. Outermost layer of the muscles, the epimysium. What's the outermost level of nerves?
Epinurium.
The epineurium.
EP.
Yeah, exactly. The epineurium. So that is the warm up there. And what we're going to do today is now we kind of left our little food up at the esophagus. It had gotten swallowed. There was deglutition, kind of squeezed it down the pharynx and started peristalsis going down. And so what we're going to do for the rest of today is look at the rest of the digestive tract. We're going to look at the stomach. We're going to look at going into the small intestine. We'll see there's 3 different sections of that. We're going to go and look at the accessory organs. We've already looked at the teeth and the salivary glands, but now we're going to look at the liver and the gallbladder and the pancreas that are adding in stuff to help break down your food. Then go into the large intestine, into the rectum, into the anus. And then also talk about the enveloping of the serous membranes around all those. Again, the peritoneum, but there's different sections of it and they have different names. And where it doubles up, they're called mesenteries, and we'll talk about the different mesenteries. So that's kind of our game plan for today. Let's see here. Do I want to? I'll go there later. Okay, so let's follow our food down into the stomach. All right. So we swallow the food, heads down the esophagus via the rhythmic peristaltic contractions. At one point, and we saw the esophagus is directly behind the trachea. So remember, it was important that the trachea, the trachea had its little C-shaped cartilage, and the esophagus, this kind of muscular tube right behind it that is squishing your food down. What kind of epithelium lines the esophagus?
Stratified squamous.
Exactly. So it has stratified squamous epithelium, which is the same as you have inside your mouth, right? Inside your mouth and covering your tongue, oral cavity is all stratified squamous. As you get down into the esophagus, there are also mucus glands underneath that are kind of sending up mucus to make it a little more slippery in there. But again, a classic test question. Like one of the slides that we have that I like a lot is a slide. It's not of a human because it wouldn't fit on a little slide. I think it's from a mouse or a rat or something. But a thing where it's a slice of the trachea and a slice of the esophagus side by side. And you have to be able to know which tube are you looking at. So when you are looking at the trachea or looking at the esophagus, what would be the major giveaways that you're in the trachea versus in the esophagus?
The trachea has like a C shape, I guess.
So the trachea is going to have the C-shaped what? It's going to have
Hyaline cartilage.
the hyaline. You'd see hyaline cartilage in the submucosa, and you'd never see that in the esophagus. So the trachea is going to have hyaline cartilage in the submucosa. What else are you going to see in the trachea that you wouldn't see in the esophagus?
Ciliated pseudostratified columnar epithelium.
Totally. It's going to be lined with ciliated pseudostratified columnar epithelium versus the esophagus, which is going to be lined with stratified squamous epithelium. In the wall of the esophagus is it's going to be that muscularis externa. So it's very easy to tell one from the other once you kind of know the basic things to look for. All right, so then we go down into the stomach. So obviously the stomach has to pierce through the diaphragm in order to get out of, it's going, what do we call that kind of area between the lungs in the thoracic cavity? What's the name for that cavity?
Mediastinum?
The mediastinum. It's going through the mediastinum in the thorax, but then it has to penetrate through the diaphragm. They call that the esophageal hiatus. And when someone has, sometimes people say someone has a hiatal hernia. A hiatal hernia is if things are kind of pushing up through there, if that is not sealed well. So then there's our stomach. Esophagus leading in. On the other side, this is going to be the duodenum, which is the first part of the small intestine. So this is kind of giving you an orientation here of what's going on. What we're going to do now is look at the stomach in much more detail. Stomach. All right, so the main kind of just big part of it we call the body. This kind of floppy upper part up here, this is called the fundus. This is floppier. This part here that's feeding into the small intestine is called the pylorus. This is more muscular. This whole kind of part, this is called the greater curvature. What's going on here? This part up here is called the lesser curvature. The entrances and exits have sphincters. So again, smooth muscle that can pinch off a tube. So we're going to have a sphincter up here, and it has a few different names you can give it. So this, and I should have mentioned this part, the entrance into the stomach is called the cardia. It's called the cardia because it's near your heart, right? If we kind of go back into this picture here, the heart, actually, if you're looking at the person from the front, here's their stomach, their heart is right here too. So this entrance into the stomach is the cardia. So sometimes they call this the cardiac sphincter. It's also known as the gastroesophageal sphincter. Right, because it's gastro means stomach, so it can be the cardiac sphincter or the gastroesophageal sphincter. Down here, we're going to have the pyloric sphincter. And the pyloric sphincter is going to be regulating the digesting food leaving the stomach and going off into the small intestine. So pyloric sphincter and cardiac sphincter. What else do we want to say about the stomach? Let's talk a bit about what is the functions of the stomach? What is the stomach? What's happening in the stomach?
Protein digestion.
So this is going to be a big place where we're going to have protein breakdown. So we're going to talk about that in more detail pretty soon. What's another, we'll see protein breakdown also happens in the small intestine. What's another function of the stomach?
Chemical breakdown.
So that's kind of getting back to what Jumbo was saying, chemical breakdown, the breakdown of the proteins using enzymes. So I want like another thing as well.
Absorptions of some proteins.
So it does do a little bit of absorption, although not proteins. All your main nutrients are going to be absorbed in your small intestine, but there's some absorption.
Water and some drugs.
Yeah, so water and some drugs. It depends. Things that are lipid, like things, aspirin can actually go across in the stomach. Alcohol can go across the wall of the stomach and get absorbed into your bloodstream. So if you're doing some tequila shots, you don't have to wait for the tequila to make it all the way down to the lower parts of your digestive tract before you start getting your buzz on, because the alcohol can cross the wall of the stomach and get into the bloodstream. But it's an absorption. So it's like, maybe if I'm like from Boston, some absorption occurs, some absorption occurs. So if we're thinking about the main absorption, that's all happening down in the small intestine. And we're going to look at that in more detail in a little bit. But there's some absorption that happens in the stomach. What else is the stomach doing for you?
Chemical breakdown.
Oh, Elsa, that's what we were just saying, right? It's like that's chemical breakdown.
No, mechanical breakdown also.
Oh, mechanical. I didn't hear you. Yeah, there's going to be mechanical breakdown. No, that's good. So the stomach is a big place where you're just smushing the food. So there is actually an additional layer in the muscularis externa. So I'm going to put this here. Has an additional layer in muscularis externa. Basically this internal oblique layer. So it's got 3 layers of muscles, and what it's kind of doing is having these waves of squish that go down, and then waves of squish that come back up, and then squish coming back down, and then squish coming back up. So you just kind of think about if you're making bread, you're smoosh, flipping it around, smush, flipping it around, smush, flipping it around, smush. So basically the food is smushing back and forth because you've got these muscles in the walls. When you first swallow your food, you chew it up, you swallow it, it comes down the pie or comes down the esophagus, drops in. It kind of just piles up right here in your this fundus area.
I have a question.
Uh-huh.
Is it filled with acid?
There is going to be acid in here, and we are going to get to that.
And in the cardia, is there like a valve or is it just open?
No, it's a valve, although it can, if it has trouble staying closed, that's when you start getting acid indigestion.
Oh, okay. Thank
So
you.
we're going to talk about that as well in a few moments when we talk about kind of how the digestive tract protects itself. So normally it should stay closed so you don't have this digesting food going back up the esophagus.
Okay.
But if it does happen, that's when you get heartburn because the esophagus is not protected from kind of the acid. So it hurts. It's like, ouch, I feel that.
Okay, thank you.
Sure. So as the food drops in here, you can actually, it looks like the regular food. And it's not immediately all filled with acid either. It's like, again, if you've just eaten a big meal and you've barfed relatively soon after, I'm sure, I can't believe anybody's gotten to this age where you haven't like kind of barfed and looked at your barf. And it's like, you can recognize what you ate. You can usually see there's chunks of spaghetti or tomatoes or whatever, half chewed piece of meat or whatever. So when you first chew the food and swallow it, it just kind of lands here in this bigger, floppier top part. But then with this mechanical breakdown, it's getting smushed back and forth, mixed up with the acid and with the digestive enzymes that we're going to talk about, and starts turning into this white paste. And the paste has a name. The paste, they call it chyme. Chyme is just the name for it no longer looks like food. If you're barfing up and you just have this white goo coming out of your mouth, that's chyme. And basically as this is going back and forth and breaking down, as the food turns into this goo, it starts getting squirted out a few mils at a time down into the intestines, the small intestine, to continue breakdown. So the other big thing that your stomach is doing is it's acting like a big storage tank. Right? You can fill up the stomach. It can hold up to a gallon or so. My corner of my screen is being weird. Up. Yeah, I can't write it there. Up to 1 gallon. So that's how you can have lunch and now sit in front of your computer here, and that food is getting smushed and fed out a few milliliters at a time into the rest of the digestive tract. In fact, for when people when they're really trying to lose weight, one of the things that happens, or one of the surgeries people do, are these stomach reductions, like stapling the stomach, trying to reduce the storage capacity of the stomach so you can't just eat a big meal. You can only eat a little bit at a time and have to wait for it to continue down the GI tract before you eat a little more. But if your stomach is working right, you can just eat a big meal and fill this thing up. It can extend all the way down almost to the level of the pelvis as it kind of expands out. So storage. Other things about the stomach. When the stomach is stretched out, it's obviously stretched out. When it's not stretched out, the walls get all kind of wrinkled up looking inside. So let's look at the stomach. We're going to look at it kind of cross section now, or open. If we look inside the stomach, you've got all these kind of folds. So walls wrinkled, wrinkles and folds. And they have a name, they're called rugae. Excuse me. We have pictures of this that you can see later. But so if you cut the stomach open and you look inside, the wall will look all kind of wrinkled up. So, and obviously if it's full, those rugae are going to get stretched out. And then the walls of the stomach are filled with all of these little things called gastric pits. The mucosa goes down into these little gastric pits with these different gastric glands underneath. So let me draw this this is going to be the wall of stomach. This is the lumen here. We got simple columnar epithelium. Then it goes down. And then there's all these glands. So we have a gastric pit. Basically kind of little hole going down. And then we have these gastric glands that are making stuff, and we'll talk about what they make in a second. All right, so this is kind of the outside. And again, you'd have the muscularis and all that stuff out beyond that. So gastric glands, there are a few types of cells you need to know that you find down in the gastric glands. So cell types. One is we have what are called parietal cells. And these are the ones that make the hydrochloric acid. HCl. HCl just means, maybe I should spell it out. And then they also make something called intrinsic factor, which we're going to talk about. There's also these things called chief cells. And they make something called pepsinogen, and we'll talk about that in a few moments. And there are also what we call enteroendocrine cells. So let me talk about each of these in turn. So first, hydrochloric acid. The lumen here is going to be very low pH. pH around 2. That's very acidic. This is doing a few different things. One is it's making it very inhospitable for bacteria and stuff that are dropping down and you're swallowing stuff. Two, the acid, it helps denature the protein. So one of the, when we talk about the protein breakdown in the stomach, the acid doesn't actually break down the protein, but it does denature. It kind of unwinds it and makes it easier to break down for the enzymes to work. And it's not hydrochloric, good God, hydrochloric acid. Hydrochloric acid, it also is going to create the optimal pH for the enzymes. The pepsin that breaks down the protein works in very low pH. It won't work in high pH. So hydrochloric acid is doing a variety of things. Intrinsic factor, this is needed to absorb vitamin B12. So this is one of the non-intuitive things about your stomach, is this ultimately becomes the only totally essential, non-replaceable job that the stomach can do. If you didn't have a stomach, in fact, like I said, sometimes for people who are trying to lose weight, they'll basically do major stomach reductions. If you don't have the stomach, you're not going to have the storage, but you could just have to eat little bits at a time. If you don't have the stomach, you're not going to have this thing that's doing protein breakdown, but it doesn't matter so much because there's plenty of protein breaking down enzymes going on in the small intestine later. So the small intestine also breaks down protein. The only thing that you definitely need the stomach for is to make this intrinsic factor to absorb B12. In fact, with people who have those stomach reduction surgeries, you need to get shots of this because it's necessary for lots of things, including making blood cells and things like that. So before we get to the, let's talk about in the lumen here of the stomach, you're going to have the acid and enzymes that are designed basically to dissolve animals into building block protein and amino acids. So how is it? So you've got this gastric juice here in the lumen, and it's designed to turn a cow into goo, right? The whole point of this is you're chewing up some other animal and you are dismantling its body into building block molecules. So how do we make sure that we don't just dissolve ourselves from the inside? Because we've got this corrosive brew here that's designed to dissolve mammals just like us. Anybody?
Can you say that again?
So the digestive juices here inside, this is the lumen of the stomach here. And these digestive juices are designed to digest protein, basically to take a mammal, take a cow and dissolve it into goo. It's basically the whole point of these enzymes and acids here are to take the flesh of a mammal and dissolve it so you can break it down and digest it and absorb the nutrients.
Isn't the stomach covered with like connective tissue?
So the connective tissue would just be more stuff, more protein that would break down and dissolve.
Mucus. No.
So yeah, so one of the things is there's a thick protective layer of mucus that is going to be between the digestive juices and the actual cells. So there's this protective mucus layer. So that is going to be one way that you protect from digesting yourself. Another way that you protect from digesting yourself is that the enzymes aren't actually made in their active form. So if we go back down to where I'm talking about the chief cells making pepsinogen here, pepsinogen is not the active enzyme that breaks down protein. Pepsinogen, once it hits the acid, becomes pepsin. And this is my protease. This is the thing that breaks down protein. So these cells down here that are making the enzyme is not active. The enzyme is not actually able to break down protein when it's getting made. It's because it's pepsinogen. It's this precursor form. And it travels up, it goes up. It's not going to damage your cells because it's not the active enzyme. But then it ends up in the acid, in the low pH up here, and kazing. Now it is activated, and now it's able to start dissolving animals. So, but when it was made and when it's transported up the little pits and stuff, it's not in the active form. So that's another way that you keep from digesting yourself, is you make the enzymes in an inactive form and they don't actually turn on until they hit the special pH. In the stomach, it's a very low pH. In the small intestine, it's actually an alkaline pH. So it's, it's different, but it's still the same idea that you have to hit this specialized environment for the enzyme to turn on, to activate. Next, there's one other thing that we can talk about in terms of protecting yourself, and it's that your life expectancy as an epithelial cell in the digestive tract is really short. Even with the mucus and with these precursor versions of the enzymes, the cells do get damaged, and you're constantly having to replace them. So there's a very high turnover of cells in here. I don't know if how can I do that? I can just say, very high turnover of epithelial cells. So all of these cells here, they're wearing out. And basically what happens is, like, as the cells wear out, they actually get pinched off and just popped up into the lumen. They just become more stuff for your body to break down and digest. You actually digest, part of what you're digesting in your digestive tract is actually the worn out cells that are, like, done, that have been lining your digestive tract. And you've got new cells migrating up from the protected pits to take over and replace those. We're going to see this in the intestine as well. There's going to be these intestinal crypts where you have cells migrating up from below to replace the cells that are wearing out on the lining there. So those are like three things that are all at work. You've got this protective mucus. Maybe let me do a different color. Protective mucus protecting you from digesting yourself. You're making your enzymes in this inactive form, so that protects you from digesting yourself. And then you've also got just this high turnover of epithelial cells, and that is helping deal with the fact that the cells are getting worn out by this corrosive environment. This is part of the reason why when people are taking chemotherapy, it often causes digestive problems. Most drugs that are used for cancer are targeting cells that have rapid cell division. And that's one of the things that distinguishes cancer cells from most other cells in your body, is that they're rapidly dividing. So most of these chemotherapy drugs will kill cells that are rapidly dividing. One place you find cells rapidly dividing are in your hair matrix. That's why your hair starts falling out. You're damaging your hair follicle or your hair matrix cells. You're also damaging your lining of your intestines. So it actually can mess with your intestinal function when you're taking these drugs. People start feeling really sick So that's kind of the thing about gastric pits. Gastric pits, they've got parietal cells making hydrochloric acid intrinsic factor, chief cells making pepsinogen, and then enteroendocrine cells. What do you think they are doing? Just from their name. Endocrine. You guys.
Endocrine cells within the digestive tract in the stomach.
Yeah, and what do endocrine cells make?
Hormones.
They make hormones. So one of the things that we're going to be finding in our study of the digestive tract today is that a lot of the control is done by hormones. These parietal cells get the message to secrete hydrochloric acid in response to a hormone called gastrin. There are all sorts of hormones that are involved in the control of your digestive tract. We'll talk about a couple of them. In this class, we don't go as deep into it, but these enteroendocrine cells are making, for instance, this hormone gastrin, which is talking to some of their neighbor cells and telling them to start secreting acid. So it is important to realize that the digestive tract is making hormones that help control digestive processes. We talked about endocrine glands more formally, but those were dedicated endocrine glands. There's lots of hormones that are not made by dedicated endocrine glands. Here's hormones made by your stomach. We're going to see more in a few moments made by your small intestine. Cell estomaco. All right, let's back up a little bit. Here's our stomach again. The stomach, again, is coming through the diaphragm here. Right, the esophagus is just covered in dense connective tissue, right? That was what we called adventitia. That's covered in adventitia. Once you poke down into the abdominal cavity, most of the stuff is going to be enveloped in the peritoneum, in that serous membrane that surrounds the digestive organs, including the stomach, including the liver. Now let me put the liver up here. It's right underneath the diaphragm. This is the diaphragm. One of the mesenteries that you should know goes from the liver to the lesser curvature of the stomach. It's called the lesser omentum. There is another mesentery that hangs off the bottom of the stomach and actually goes down kind of like a big apron and kind of comes back up and kind of anchors on the transverse colon. This, and again, I know this picture is probably a little confusing, so we're gonna see this from many different angles before we're done today. But this is called the greater omentum. So just kind of introducing the ideas here of these mesenteries enveloping things and getting kind of stretched out between different organs. There is also one that actually goes right between the lobes of the liver and attaches the liver onto the diaphragm. It's called the falciform ligament. And actually the very bottom of the falciform ligament has, let me maybe do it in green. On the very bottom of the falciform ligament is the remnant of the umbilical vein. So this is called the round ligament. It's the vestige of the umbilical vein. Again, if we were in lab, we actually have the cadaver dissected really nice where we actually cut and left his belly button intact. And you can actually see this fibrous cord going from below the liver there, but on the bottom part of the falciform ligament, and it just connects to his belly button, the backside of his belly button. You know, because when you are in utero, obviously there's just a blood vessel, the blood's returning for the placenta coming back into the liver there. But once you're an adult, you know, that vessel doesn't just dissolve, it turns into just kind of this connective tissue, which you can still see. Alrighty then. If you think if there's anything else about the stomach. I think that's it for the stomach. Let's continue. Or actually, you know, I could. Yeah, let me do this. I'm gonna show you this from the viewpoint of the PowerPoint so you can actually see a prettier picture of it. So yeah, I'm gonna put all of these I'll put these all up on the web after class. The basic idea here, go pointer. You know, here's the diaphragm here. Here you can see the serous membrane surrounding the liver. The falciform ligament is that little thing connecting the liver to the diaphragm. Then it folds into a double layer, into a mesentery, and now it's connected to the top of the stomach. And then it envelops the stomach. The visceral peritoneum is just single layer on either side of the stomach, but then it joins up into a double layer again. This is now the greater omentum hanging off the bottom of the stomach, going down like an apron in front of all of your intestines, coming back up and anchoring on the transverse colon here. This is like the back part of your large intestine. Here you can see most of the intestines are all hanging off the back of the body wall in what we call like the mesentery proper. There are just all these double layers of serous membrane that goes down, wraps around, comes back up. You know, so your intestines are not just like a garden hose shoved into your belly. They're actually supported by these mesenteries. These mesenteries support the blood vessels and nerves and stuff that are feeding the intestines as well. Not everything is wrapped up in mesenteries. There are some things that during development, they end up migrating back behind, and like the duodenum, the first part of the small intestine is actually retroperitoneal. It actually goes back behind the peritoneum, but then it pops back out. As soon as you're in the jejunum, you're back into the peritoneum. We've already talked about how the pancreas is sitting here, retroperitoneal behind the stomach, right? Again, if we were doing the little piggies, you would take the stomach, move it out of the way, and it's like, I don't see the pancreas, until you take your little needle and then rip away all this shiny, slippery membrane, which is the peritoneum, the serous membrane. And it's like, oh, there it is. It's just sitting there behind. This is just kind of showing the same idea. You know, peritoneum is kind of just wrapping around all sorts of stuff, wraps around the spleen. But here we have like the kidneys are also retroperitoneal. So when you're thinking about the peritoneum, most things are enveloped in it. That means everything has this slippery coating on it. So everything that touches something else has got this really slippery bearing surface. So nothing is getting friction. So that, hopefully this makes the peritoneum make more sense to you all. And here's the bladder, which is kind of stuck down below. The rectum is also retroperitoneal here. So does this make sense? Okay. Yeah, so intraperitoneal just means they are within the peritoneum. They're just enveloped in this membrane. And there is a, pedantic thing. Secondarily retroperitoneal is when things during development lose their mesenteries and move behind, but you don't need to know if something's secondary retroperitoneal. What's next? Let's see. So next we're going to get down into the small intestine. So our stomach smushing the food, turning it into chyme, into this white goopy paste. And then a few mils at a time, it's squirting it out through the pyloric sphincter into the next section. Oh, actually, I'm going to go back to the stomach for one more second. It's kind of going back to what Sakai was talking about a little earlier. Actually, I have to redraw the stomach. So we talked about how the stomach has mucus and it has all these different things that help protect it from digesting itself. The esophagus is not going to have that. The esophagus is got protection against friction. The esophagus has stratified squamous epithelium, so it's not going to get injured from friction of food coming down. But it normally doesn't have to worry about digestive juices dissolving meat, right? So if you do have, here you've got this corrosive brew of digestive enzymes, designed to dissolve meat. If it does end up getting up into here, it's gonna burn. It's gonna start damaging the walls of the esophagus. And that is, heartburn. They call it heartburn because, the cardia, the place where this is near your heart. So it kind of feels like your heart is hurting, but it's actually your esophagus is in pain. So they also call it like GERD. It's like, it seems like a weird word. Esophageal reflux disease. Reflux just means kind of bubbling back up. So obviously, this can happen because the sphincter is weakened. It's worse like if you're lying down, if you've just, or if you're really full, there's a lot more back pressure to push it up. If you're lying down versus sitting up, there's more of a pressure. Even which side of you're sleeping on in bed can make it more or less likely that you're having heartburn, because of the fact that your stomach is on your left side, and whether you're squishing it sleeping on that side or sleeping on the other side, so the stomach has a little more room to breathe, not get compressed, I should say. Worst case, if you actually start not just irritating, but burning a hole in the wall of the digestive tract, then we have what's called an ulcer. And you can have different kinds of ulcers. You can have esophageal ulcers where you start actually damaging the wall up here. You can have peptic ulcers where you're eating away at the wall of your stomach. They can get so bad that you can have perforating ulcers that go all the way through even. You can have duodenal ulcers where you are getting damage to the beginning of your small intestine. So ulcers are where you start eating away at the wall of your digestive tract. They used to think, it's more just like stress or something. And now they found that there's actually a bacteria that's often associated with ulcers. It's called Heliobacter pylori. It's like an interesting story. At first, the guy who was suggesting that this bacteria was an important part of a lot of people getting ulcers, people thought he was kind of an idiot or kind of a wingnut, and he did the critical experiment where he made a batch of a culture of this bacteria, and he drank it, and within days he started getting ulcers all over the place. And he proved his point, and now at this point they actually will often give you antibiotics as part of treatment for ulcers, because it's clear that often that these bacteria are playing a role in the process there. So, but yeah, heartburn is just having that kind of acidic corrosive mix getting up into the esophagus where the cells are not protected from the chemical breakdown. Okay. Let us now we can have the food is getting squirted out in little bits into the duodenum. This is the first part of the small intestine. It kind of acts like a mixing zone. So it's retroperitoneal, if you care. It actually dives back a bit and then it's going to pop back out. It's very short. It's only like 10 inches long. I think duodenum, it comes from a word, it means 12 fingers, because it's about the length of 12 fingers, and side by side. It's kind of a weird name for it. So it's a place where your stomach is adding in its mix. We need to now add in the pancreas. The pancreas we've talked about the pancreas as an endocrine organ making insulin and glucagon in those pancreatic islets of Langerhans, but we also mentioned that it has acinar cells that are making digestive enzymes and juices. So we're going to have the pancreatic duct, which is going to come on. Enter in. And it's going to dump what is it going to dump? Let's talk about what the pancreas makes. So the pancreas makes two things. It makes bicarbonate juice. So this is, bicarbonate is basically baking soda. This neutralizes acid. In fact, if you have acid indigestion, old school, you can just like mix up a little bit of baking soda in water and it neutralizes the acid in your stomach. So bicarbonate juice, as the chyme is coming down from the stomach, this bicarbonate juice will neutralize that acid and actually make it alkaline. So it neutralizes the acid. And then 2 makes enzyme juice. And it makes lots of enzymes. So it makes proteases. These break down proteins. So it's got, it's got proteases. In this case, it's not pepsin, it's something called trypsin. It makes lipases. These break down fats. Makes amylases. There's pancreatic amylase, which break down starch, break down carbohydrates. Nucleases break down nucleic acids. So basically, it makes enzymes that can break down pretty much anything that you need to break down. So it makes all sorts of enzymes. This is why I said the stomach is not essential for protein breakdown, because the pancreas is making enzymes that break down proteins as well. The other thing that feeds into here, let's add in the liver. We're going to add in the gallbladder. And some little ducts here that we're all going to figure out in a few moments. All right, so let's talk about bile briefly. Bile salts. So breaking down fats is particularly tricky because fats don't dissolve in water, right? Let's say you just ate a big pat of butter or drank a shot glass of olive oil or something, right? Oil in water separates. So the oil stays as a big glob. It doesn't, let's say this is my oil here. And I might have enzymes. In fact, I do. I said lipases. I have little enzymes that can try to break down the oil, the fat, into building block molecules and the fatty acids and glycerols and stuff. But it can't get at most of them. Most of them are protected deep inside the big blob of oil. So, and you could mix things up into lots of, let's mix it up. So we've got lots of little oil droplets, but bing, you know what happens, they separate. So we need to have some way that we can mix this oil up and keep it from regrouping so the enzymes can actually get at the oils and break it down. So that's where bile salts come in. Bile salts are, they call amphipathic. They have one side likes water loving. We call hydrophilic. Whoops. One side is, lipophilic. I'll just say lipophilic. One side likes fats. So one side likes water, one side likes fats. So whenever you get like a little droplet, the bile kind of gets in there. The lipophilic, the fat-loving part goes towards the fat drop. The water-loving part goes out towards the water. And that isolates this Y so he can stay intact as an independent little droplet. So we call this an emulsifier. It doesn't actually break the fat down, but it keeps it from regrouping back into one big blob.
So it's like a soap.
Yeah, exactly. It's like a soap. In fact, like a detergent it's exactly like a detergent. This is how your detergent works. Like when you have a greasy plate and you just put it under the faucet, the water just, goes over the greasy plate and the grease stays on the plate. But when you use your detergent, then as the oil gets kind of moved around and it gets kind of sequestered by the detergent like this, now the little droplets can now get into the water and go along with the stream and go down the drain. So this is how detergents work as well. So this is exactly, and this is also, what they put like in, if you got Bernstein's, like one of those pre-made Italian dressings, like if you make your own like oil and vinegar and you shake it up, it separates immediately. But if you get something with emulsifiers in it, there's lots of different emulsifiers. Egg whites actually can be emulsifier, or egg can be emulsifier, or carrageenan is a classic emulsifier that people use in food preparation, which is like from seaweed. But anyway, the bile salts emulsify the fats, and now my enzymes have way more surface area to work. So now the enzymes are way more efficient. So I can break down fats way more efficiently as long as I have bile salts. The bile salts don't break down the fat, but they really help the enzymes do their job by increasing the surface area to work on. So if we go back here, that's the whole point of the gallbladder. The gallbladder stores bile, stores and concentrates bile. And so it adds it into the mix as well to help the lipases from the pancreas do their job. What happens if you have your gallbladder removed? It's actually not an uncommon thing.
You can't have a lot of fatty foods and fried foods.
Yeah, exactly. You have to be more conscientious about eating fatty foods because you're not going to be as efficient at digesting fats as you used to be. So let me look at the time. So the gallbladder, I'm saying it stores and concentrates. It doesn't make the bile. The bile is actually made in the liver. So the bile is made in the liver and then moves in through what are called these little hepatic ducts. And so the bile goes on. Another sphincter we need to add in here. The hepatopancreatic sphincter of Oddi. So the hepatopancreatic sphincter of Oddi is controlling whether or not these juices from the pancreas and the bile from the gallbladder are let into the duodenum here. And there are more words associated. There's the hepatopancreatic ampulla, which is the kind of more swelling area where these things are entering. The duodenal papilla, where you can see a little nipple-like projection. You won't be tested on those, but you do need to know the hepatopancreatic sphincter of Oddi. And so if the digestive processes don't need the bile at the moment, what happens is the liver is making the bile, and the bile is going through this hepatic duct, but it's not going to go. This is called the common bile duct here. And instead of going down the common bile duct to get into there, it's going to back up into the gallbladder through what's called the cystic duct. So the basic idea here, maybe you just do it. So liver is making bile. Assuming that this sphincter of Oddi is closed, the bile is just going to go up the cystic duct, up into the gallbladder where it's stored and concentrated. When your digestive tract needs bile, it sends a message that we're going to talk about in a little bit to tell the gallbladder to squeeze, which will then squirt the bile down into the small intestine here through the common bile duct. So hopefully that makes sense there. Whoops. Whoops, no, where was I? I was over here. So that was just kind of showing up here in more detail. So bile made in the liver, stored in the gallbladder. This, maybe, so the last thing I'll do before we do our break, let's talk about the control. How does the pancreas know that it's time to secrete bicarbonate juice or secrete enzyme juice? How does the gallbladder know that it needs to contract and supply bile? And it turns out that it is hormones that are doing the control here. This was one of the, I think this might have been the first place where physiologists first discovered hormones. They were trying to figure out how the heck does the pancreas and the gallbladder know that it's time for them to secrete when the food gets into the small intestine. And I think they probably did it on dogs or something. They like cut everything they could see that connected them, every nerve. There was like no physical connections between the digestive tract and the pancreas and the gallbladder. And still, as soon as the food ended up in the small intestine, the pancreas and the gallbladder start doing their thing. And what they realized was that the message was not by a direct connection at all. It was because the duodenum was releasing hormones into the bloodstream. And then those hormones then were detected by receptors in the pancreas and the gallbladder, and that's what triggered them to do their secretion. So I am going to introduce the two hormones that you should know that are involved. There's lots more than two that are involved in digestive control, but I think it's worth learning a couple of them just so you can get a hit off of how important they are in the control of your digestive processes. So Some hormones released by the small intestine. One, and again, this is back in the beginning, when people first get something and don't realize there's going to be a whole bunch of similar things, they give it these names that are really generic. Secretin is the first one, because it was like, wow, we found this thing that makes something secrete. And now it's like, it's so many things that make things secrete. So secretin, hormone that tells pancreas to secrete bicarbonate juice. What is going to be the stimulus? What's going to stimulate the small intestine to release secretin? What kind of conditions would you want bicarbonate juice delivered?
When the food's going from the stomach and into the small intestine.
And what in particular do you think is going to be the quality of that food that's going to trigger the release of bicarbonate juice?
It's going to be acidic.
Exactly. So this is going to be, released in response, to low pH. So if the conditions are acidic in there, you're going to send this out to the bloodstream. The pancreas gets the message, releases bicarbonate juice, which helps neutralize the acid and make things more alkaline inside the small intestine. Another one, it's got a mouthful, cholecystokinin. But you can just call it CCK. That's its kind of more classic name. CCK tells gallbladder to deliver bile, and pancreas to deliver digestive enzymes. So this one obviously is going to be triggered by the presence of, food, digestive, the substrates of digestion. Are there fats and other things that you need to break down? Another interesting thing about cholecystokinin is it doesn't just talk to the pancreas and the liver or the gallbladder. It also talks to the stomach. So if I go back over to here, we're sending the food out into the little bit of squirt at a time down into the duodenum. If the duodenum has got food in there right now and it's saying, I need more digestive enzymes to break stuff down, that same hormone actually tells the stomach, please slow down, inhibit gastric emptying, because I've already working on stuff that's here. We're not done. So when the duodenum is releasing cholecystokinin, it's actually also telling the stomach to inhibit emptying, so it has the small intestine has a chance to keep working on what it's got already, rather than having more stuff come in. So foods that are more difficult to digest, like fatty foods, are going to make you feel full longer, because they're going to be sending back these messages to the stomach that are keeping the stomach from emptying as fast. So, different kinds of foods are going to make you stay full longer, particularly ones that are more fatty, or make you feel like hungrier quicker. So that's kind of an interesting thing as well. Okie dokie. So when we come back, what we'll do is we'll just finish up the intestine. The colon will be pretty quick. The rectum be even faster. And then we'll also get into some lab stuff. I've looked around and found some little clips from the cadaver dissection videos that will help give you a better sense of this stuff. We have a little bit of more histology to look at, but it is currently 2:06. So let's say a quarter to 3, 2:45. We will regroup and finish up the digestive tract and do some labby kind of stuff. All right, so we are going to continue talking about the digestive system here. And we will go. So we were in the stomach. Squirted out through the through the pyloric sphincter into the duodenum. And then from the duodenum, we head into the main part of the small intestine. So there's the duodenum is just like the short little mixing area. But then you have the jejunum. And then there's going to be, the other half is going to be the ileum. It's about 50-50 in terms of the length. But functionally, they're very different. The jejunum is going to be the primary site both of breakdown and of absorption of your food. This is where we're going to spend most of our time. Ileum, reabsorbs bile salts, reclaims bile salts. I don't know why my little reclaims bile salts. Hold on. Let me use the typing tool. That way it can type over there. The ileum also has a lot of lymphoid tissue that we saw as well. But the jejunum is where we're going to spend our time right now. Primary site of breakdown and absorption. And we've already kind of introduced some of the important points, which was how incredibly folded the inside of this tube is. So let's talk about levels of folding to increase surface area. Then we'll just look at a little detail about how the actual nutrients are absorbed from the lumen into the bloodstream. So inside the small intestine. Here's the big tube. The first thing that you see when you open it up, if we kind of look at it as a cross section, you'll see these kind of big kind of ridges. These are really, these are like a 1/4 inch. You can really see them. There are these big floppy ridges. They're called the plicae circularis. Yeah, oh my God, my circularis. And later on, I actually have found a video that actually has a really nice image of them, so you can actually see what they look like on a real person. And then I mentioned before, when you actually look at the tissue here, it doesn't look smooth. It looks kind of velvety, like it's got like texture. And that's because it's thrown up into millions and millions and millions of little fingers, the little villi. So, like if I was going to draw a plique circularis, one of these are also called circular folds. Let's say this is like the submucosa, and this is my like plique circularis fold. What I'm going to see is that the mucosa, there's all these little fingers on top of it. Again, this is simple cuboidal epithelium zipping up and down. These are my villi, the little finger-like projections. So that's the second level of folding. You've got these big folds, which are actually pretty easy to see with the naked eye. You've got the villi, which they're really dinky, but you can kind of notice that the surface isn't smooth. It's kind of velvety. And then the third layer that we mentioned last time is the fact that each of these cells that makes up the columnar epithelium has microvilli. So if I take this and I look at it in more kind of magnified detail, right, here's my simple columnar epithelium. Each of those cells has microvilli. This might actually be worth doing one of the, hold on, let me find, give me a second here. Yeah, here, I'm going to show you the thing from the PowerPoint here because they do this well. So here, if you're taking, this is a simple columnar epithelium making the villus. This is lamina propria underneath. This is showing those tiny little fingers going on the plique circularis. So what? I want a better picture. Here, this is showing it really well. This shows it better than I could draw it, really. This is showing the plique circularis, those big folds, those big ridges, the villi, the tiny little fingers coming up, just covering it again like a carpet, like a shag carpet. And I was saying velvety, you can also think of it like a shag carpet. This is looking in at high magnification, so you can see all the tiny little villi, just crazy amounts of increase in surface area. Plique are the big ridges, the villi are the tiny little fingers, the little shag rug. And then this is showing that each of the individual cells, columnar epithelium cells, has microvilli that give you even more surface area. So again, the people who, I'm not quite sure exactly how they calculate it, but the number they usually throw out is you have a surface area equivalent to about a football field folded up inside your intestine in your stomach there, or in your belly.
I have a question.
Uh-huh.
So does the ET that covers that, that's just simple columnar, or is it pseudo stratified
No, it's simple columnar.
with goblet cells?
Exactly. You know, there's more or less goblet cells depending on where you're looking. In the intestines here, there are some goblet cells. When you get down into the colon, there's tons of goblet cells. So when you look at the cells in the columnar epithelium, enterocytes is just the more generic term for the cells that are making the walls that absorb the nutrients across. Goblet cells are your classic ones that will make mucus. There are also other cells. There's like Paneth cells that make antimicrobial things you find down in there. This is worth looking at, this intestinal crypt. So that's this little little gopher hole here. You can see kind of little entrance to the crypts here on the floor. Those are where you have, so they have here some enteroendocrine cells, but it's also where you have the new cells migrating up that are replacing the worn out cells. So it's kind of like a reservoir of cells that are, you know, coming in from, you know, can reserve to take over for the ones that are wearing out on the front lines. The crypts, this is the more generic term. When I was learning them, they were the intestinal crypts of Lieberkühn. You got to use the umlaut. So then you have to, when you pronounce it, you have to like like Lieberkühn. So intestinal crypts of Lieberkühn are the ones where you've got new cells kind of migrating up, and there's some cells making hormones, etc. So the last thing I want to talk about while we're in here is the absorption of the nutrients across the wall. You know, the breakdown is happening just with the enzymes from the pancreas and the bile from the gallbladder and everything smushing up with the segmentation. Things are breaking down. But the last step is going to be the absorbing the nutrients into your bloodstream. And there's capillaries and lymph vessels within the villus. So let me do that. So let's make it a different color. So here's my villus. With my simple columnar epithelium. With the little microvilli, AKA brush border. So this is the lumen. This is the villus here. This is the lumen. This is where your digesting food is. And then inside, on the other side, in the lamina propria is where you have your capillaries that are going to be the place to absorb the nutrients. And there's also going to be a lymph capillary, which we're going to call the lacteal. And you'll see why we need that in a moment. So most of the food breakdown is happening with the enzymes. And actually the very final steps, there are actually enzymes bound onto these columnar cells. They're called the brush border enzymes that do the final steps of breakdown. But hold on. Whoa, shucks. I wanted to stop the video so you didn't see like the bottom of my neck, but I got rid of the other thing. Dagnabbit. Um, black. Sorry about that. All right, I will recreate this really fast. So most of the nutrients you can just carry across. Again, this is not something for this class. In physiology, we go much more into detail, the idea of transepithelial transport. You know, you'll have maybe some glucose, and there is a whole cool way with different membrane proteins where you bring it in and bring it across and spit it out on the other side. And things like sugar can get absorbed directly into the bloodstream. So sugars, I'll say more generically, I'll say carbs, and also amino acids, which are the breakdown of proteins. These can get absorbed directly into the bloodstream, absorbed into capillaries. So you just have to bring them across the wall, across the columnar epithelium, and then they can just get into the bloodstream. And then they're actually going to go off into the, you know, where do they end up next, actually?
The hepatic portal system.
Yeah, the hepatic portal system. You know, this is, there are a lot of drugs that are not going to be active orally, because if you absorb something through your digestive tract like this, the first place it goes is to the liver, where there's all sorts of enzymes that often deactivate things. It's all about detoxifying things and breaking things down. So there are a lot of drugs that don't work if you try to do them orally, but they will work if you get them directly into your bloodstream across like the nasal mucosa or sublingually under your tongue or something. So there are certain drugs that you can't take orally, because if you absorb them through the capillaries in the intestines, they're going to take a detour in the liver and can get affected by the enzymes in the liver, you know, which is not going to be the case if you get the things into the bloodstream directly with a needle, or again, across like the mucosa under your tongue or in your nasal cavity, or rectal suppositories, right? You know, it's another. So there's different ways to get things into the bloodstream without absorbing it directly across here. So carbohydrates and proteins, you can just absorb. Fats are a different kettle of fish, though, because fats don't dissolve in water. In order to have fats float around in water, you have to make these more complicated structures called lipoproteins. Lipoproteins, lipid is the fat. Protein is, you have an outer coat that's protein. So a lipoprotein has a protein part and a lipid part. And as an entire unit, it will float in water, because the protein part is hydrophilic, will be in water. And the lipid is kind of stored in the middle to be transported around. So lipoproteins are how you transport lipids, fats around in your body. Again, we're not getting into the bigger picture of high density lipoproteins, low density lipoproteins, and all that transporting fats around. The specific lipoprotein we're going to look at in absorbing fats in your intestine is called a chylomicron. So I'm not even sure why it got that name. That is the name of the little lipoproteins that are assembled by the epithelial cells in your intestine. And basically what happens is, as the fats break down into the building blocks, they get taken up and they are assembled into chylomicrons by the columnar cells here. So fats are assembled into chylomicrons, which then are spit out on this other side here. So this is basically the fat assembled into this little thing. And now it's too big to go into a capillary. So you can't put it directly into a capillary. So what happens is it can get into a lymphatic. So the chylomicron gets absorbed by the lacteal. So fats are going to have a very different pathway to get into your bloodstream. You've got fats are getting broken down, assembled into these little lipoproteins called chylomicrons. The chylomicrons are too big to absorb into a capillary, but we talked about lacteals with those little flappy endothelial cells. So you can actually get it into the lacteal. And those are all going to dump down into the bottom of the thoracic duct, which is called the what?
Cisterna chyli.
The cisterna chyli, exactly. So all of these are now kind of dumping into the cisterna chyli. And then from where do we end up? Where does the cisterna chyli and thoracic duct ultimately drain into?
Brachiocephalic.
Yeah, exactly. Just into the brachiocephalic vein. And now the fats are in the bloodstream. But they did not go through the liver. They actually went straight into, took this detour through the lymphatics and then into the bloodstream. So like, on a test, you should know the difference between absorbing fats versus absorbing other kinds of nutrients. Most nutrients you just take across the epithelium and they get absorbed into the capillaries and end up going to the hepatic portal system. Fats, on the other hand, get assembled into these lipoproteins, get absorbed by the lacteals, which are the lymphatic capillary, and ultimately end up into the venous circulation through the thoracic duct. So that makes sense? Okay. So I think the ileum is not so much about absorption. And when we look at the ileum on the cadaver, you're gonna see it has much less folding. It's not, because when you want to have a lot of absorption, you are going to want a lot of surface area. Since the ileum is not about major absorbing nutrients, it's not going to have such elaborate folding. All right, so now we've got stomach, small intestine. So the final part is going to be the large intestine. Also known as your colon. Things about the colon. It's called large because of the diameter, not because it's long. It's actually pretty short. It's got this kind of pouchy beginning, then it goes up, then it goes over, then it comes down, then it makes a little S-shaped thing, then there's the rectum, and then there's the anus. So let's talk about the different sections and what's going on. So this first kind of part where it starts, this is called the cecum. It's kind of this pouchy area right here. In fact, the little entrance from the ileum into the cecum, this is called the ileocecal valve. depending on actually on the, it's often gone, but hanging off of the cecum is where you find the appendix. The vermiform appendix is its official name. Vermiform meaning worm-like. And in the old days, they used to think, oh, it's just some vestige, we don't use it for anything. Now it seems it might be an important place to have bacteria that will kind of reseed your intestinal flora. You obviously can get along without it because a lot of people have them cut out, but it's not so clear that it really doesn't do anything useful. It's filled with lymphatic tissue. If you look at it a cross section, it's just full of lymphatic tissue. So then we have just ascending colon. So this is called the hepatic flexure. Also called the right flexure because it's, kind of right near your liver. The transverse colon. That's the one that's just going across. What do you think this flexure is called?
Descending.
Left.
So this is the descending colon, but the flexure.
The left splenic flexure.
Exactly. The left or splenic flexure because it's near the spleen. Then we have to do this little zigzag because your anus is in the center of your body. So there's what's called the sigmoid colon. So those are the basic parts of it. We'll come back to it in a moment. Let's just finish up with the rectum and the anal canal here. The rectum, so one of the things that happens, the bacteria in here make gas. Even if they're not making so much gas that you're feeling like, whoa, I got a lot of gas. They're still making gas. And you want to be able to have that gas escape without splattering out feces. So the rectum is kind of this little valve, valves to separate flatus from feces. Right, it's kind of nice that you can let the gas escape without spraying poop out of your butt. The anal canal. The anal canal, there's a couple of sphincters, just like we saw with the urinary system. Actually, no, we haven't done the urinary system. That you're gonna see in the urinary system. So there's gonna be an internal anal sphincter. Which is going to be smooth muscle. And then we're going to have a, whoops, I didn't mean to do that. And then we're going to have an external anal sphincter. Which is going to be skeletal muscle. So the basic idea here is as the rectum and lower part of the colon fills up with feces, they get stretched out. And that triggers a reflex. It's a spinal reflex. As the rectum and the lower part of the colon are getting stretched, it will cause the internal sphincter to relax. Smooth is involuntary. that puts pressure down on the lower part. And that's when you feel the urge to defecate. It's like, I gotta poop. But luckily, the external anal sphincter is voluntary. So you have a choice of whether or not you're going to relax that one or not. So internal one is just going to relax automatically as things fill up. And as the feces kind of pushes a little further down, you'll feel that as the urge to go. And then you can choose, is it appropriate time and place? Let's talk a little more about the colon. Again. So what is it doing? Functions. What does it do?
It's digesting, tough to digest stuff.
So not really, although I can say, like, there is some breakdown of, like, part of the reason you have gases and stuff, like cellulose is something you can't digest. You don't have enzymes to break down cellulose, but some of these bacteria can. And as they break them down, that's part of where that gas comes. That's why, like, beans, things that have a lot of fiber are more likely to give you more gas. So there's a lot of bacteria in here. You know, exactly what those bacteria are doing, it's becoming a more and more fascinating story. It's clear now that those bacteria are actually important for the proper development of your immune system. In fact, there are some, like, diseases now that they have actually found that they can cure by doing what they call fecal transplants, kind of changing the type of bacteria that are found in the colon, and it kind of makes everything all of a sudden work okay with the immune system. The bacteria are doing some metabolizing of stuff to make vitamins. So there's bacteria make some vitamins that you can absorb. I think there's some B vitamins, K and stuff. So you can absorb those here. The bacteria are important actually for all sorts of stuff. They actually make things that, you know, get absorbed into your bloodstream. They've actually shown now that, like, your brain doesn't develop properly without having the proper bacteria in the gut. And they've actually shown now, like with these rat experiments, you can feed rats the exact same diet and they have these different set point weights. And if you swap out their gut bacteria, the two rats will now swap their set weight, even though nothing else is changing. They're still eating the exact same diets and everything. So there's something about the bacteria in your gut that affect your metabolism and stuff and where your weight is given the amount of food you eat. There's evidence that the bacteria actually tweak your nervous system in ways to make you crave the kinds of things that they like to, the kind of substrates they like to digest. So they're kind of like playing you a little to eat the stuff that they want to have coming down the line into your digestive tract. So it's really trippy. It's very trippy. The more we, like, about half the volume of your poop is not waste product from your food. It's actually just bacteria that are just growing and living inside your intestines. You know, people often talk about there's way, way, way more bacterial cells in your body than there are human cells. So what's another really big thing? So the bacteria definitely do lots of stuff, including making some vitamins. But the biggest thing that happens here is water reabsorption. you've got all of these juices pouring into your intestines, all these digestive juices. And so all of the digesting food is this big sloppy slurry. And you need to make sure you reabsorb most of that water and kind of, with, we say, compact the feces. So you don't, dehydrate. In fact, what do we call it if you don't pull enough water out of the feces before it leaves your body?
Diarrhea.
Yeah, diarrhea. And diarrhea can be deadly. If you can't stop it, it can cause like really dangerous dehydration. What's the opposite? If you pull back too much water and the feces starts getting really hard and you can't make it move anymore.
Constipation.
Yeah, so constipation is like the opposite, is over reabsorption of water. Part of why fiber, roughage, cellulose is good in your diet is it provides kind of bulk for the feces, and it holds, so that bulk gives you something to squish and push along to make sure that the feces doesn't stay too long and you suck so much water out that it gets too hard. And it also it makes it kind of almost like a sponge to hold some water in to keep it moist so it stays a little pliable. Yeah, if it gets too much taken out, it can actually just become completely rock-like. Something some of you might end up doing in the future is dealing with an impacted bowel. It's something that's kind of nice as teaching this stuff. I never have to go in and have to chip rock-like feces out of somebody so they can like kind of start moving again. So water reabsorption, bacteria making some vitamins. There's a couple of things we can talk about anatomically to just tie this all up. The longitudinal layer of the muscularis externa is reduced. Instead of being all the way around, it's in strips. So you actually see, it looks like a strip going along the outside. It's called the tinea coli, means the ribbon of the colon. And it's basically the longitudinal muscle of the muscularis externa. And then because this tightens things up, it actually makes everything pucker. So the puckers that you see in the colon, they have a name. They're called the haustra. And finally, I just love this word. Hanging off of the tinea coli and the mesentery are these fat droplets, and they're really obvious. You can just see them all over the place. There's these fat droplets hanging off. And those fat droplets are called the epiploic appendages. So those are the epiploic appendages. All right, so I think I'm gonna go into the PowerPoint and just review a few things quickly with prettier pictures, and then we can start like our lab part. Okay. This is just pretty photomicrograph with crazy colors showing the little microvilli. So try to remember this idea of, I'm going to be showing you pictures of actual cadavers in a little bit where you can just see this big apron of fat that you can then pull up and see the intestines underneath. So again, remember you've got this mesentery, it's a double mesentery, 2 double layers going off the bottom of the stomach, hanging like an apron, coming back up to the colon. And if you lift it up, you'll see all the intestines sitting underneath it. Again, this is showing the lesser omentum, that mesentery that goes from the liver to the stomach. This is showing what the greater omentum looks like. It's really just this huge curtain of fat. I have to say, like, when I, seeing enough of the kind of more overweight cadavers with really just tons of this fat inside has kind of made me always think twice before I like eat something I don't need to eat, because it's like all the stuff that's inside of you. Falciform ligament, you can kind of, this is not a great view of it, but it's this little ligament. It's actually a mesentery, but they call it a ligament, that is kind of dividing the right and left lobes of the liver, the little round ligament, which you can't see really well in this picture either, right underneath it. I'll show you other views of it in a few moments. This is kind of just showing these mesenteries that all of the intestines hang off of. You don't need to know transverse mesocolon or sigmoid mesocolon. They're just more of these mesenteries, but those aren't ones you need to know. Should definitely know this. Peristalsis versus segmentation. Oh, so this is nice. This is showing the rugae better. Again, if you just look inside the stomach, you'll just see these kind of all these ridges and folds like this. And again, they stretch out as the stomach gets full. You can also see there is 3 layers of muscle. There's the longitudinal layer, the circular layer, but then the unique oblique layer. It's the only part of the digestive tract that has this extra layer of muscle in it. This is just kind of showing the idea in the gastric pits, kind of like these little gopher holes going down, and then at the bottom of the gopher holes are all of these glands, the chief cells and the parietal cells and enteroendocrine cells. You are not responsible for recognizing like fundus versus pylorus or something. You should know where, what those sections are of the stomach, but not histologically. So don't get worried about recognizing what part of the stomach am I in. If you saw this, you would know you're in the stomach because it's got pits going down into glands, but don't worry about if you're in the fundus or the pylorus or the body or whatever. Oh, this is showing that here's the gallbladder going into the cystic duct. There's the common hepatic duct going into the bile duct and the pancreatic duct coming in together here at this hepatopancreatic sphincter of Oddi. Coming into the duodenum. So you should know that. For the liver, there is a right and a left lobe, and there's also one little caudate lobe behind, but nothing, you don't need to know anything beyond that. We just talked about all this. This is just showing that there's this stretch, reflex. As you stretch out your rectum and colon, it's going to relax the involuntary smooth muscle, and you're going to feel the urge. Under the microscope, the colon has these crypts, these kind of, again, like gopher holes, but they're just lined with a whole bunch of goblet cells to make everything really mucousy and make things slip and slide. Remember, these are the ones I showed you last time where if you cut them cross-section, they look like beautiful flowers. Because you can see the little lumen with the goblet cells all around. You should be able to recognize, what part of the intestine or what part of the canal you're in. Esophagus is going to have stratified squamous. Nothing else will have that. The stomach, you've got these pits going down into these big crazy glands. Small intestine, you have villi, you have the upward going fingers rather than the pits going down. Right, so think about fingers going up versus gopher holes going down. Same thing in the colon. The colon also has gopher holes, but instead of having crazy glands underneath, they have just a whole bunch of goblet cells making mucus everywhere. And here's esophagus and bigger. And if I do put this, I have some pictures of this that are kind of pretty. I'd make it very clear that it's a finger versus a duct or a crypt. This is just showing that falciform ligament again. This round ligament underneath, that's the remnant of your old umbilical vein. The liver is huge. People always get surprised at how big the liver really is. And you can see it sitting here in the rib cage. It's huge. And it's globbing right around the vena cava, which you'll see in the little videos we're going to see in a few moments. When they want to cut the liver out of the body, you basically have to cut the vena cava, a little chunk out of it, because the liver is kind of surrounding the vena cava there. You should know the cystic duct, which takes you from the gallbladder to this junction, the main bile duct, which is the one that goes to the actual intestine, and the common hepatic duct, which is the one that is bringing the bile in from the liver tissue. Don't worry about the caudate or the quadrate lobe. You are not responsible for those. And I think in the interest of time, even though this stuff is kind of cool, we're not gonna do the microscopic structure of the liver and the little liver lobules. The basic idea we have covered. We've talked about the liver is filtering your blood, recycling blood cells. So you have blood from the hepatic artery coming in, getting processed by the hepatocytes. We know that it's processing your food you're absorbing from your digestive tract. So you have blood coming in from the portal vein, coming through, getting processed by the hepatocytes. So that's kind of the two main things that are happening. And at the same time, the liver cells are making bile, which is going into these little bile ducts that are going to go off to your body or go off to your gallbladder. So let's do like just liver in a nutshell though. Liver does so many things. It's worth just for one time just saying, what are all the things the liver does? So we've talked about a bunch of them. What are things that the liver does?
Filters blood.
Filters your blood.
Stores glucose.
So filters blood, it stores glucose and other nutrients. Like the liver is like if your sugar is crashing, the liver is the one that is going to release more glucose into your bloodstream. It also stores all sorts of other vitamins and stuff like that. What else?
It secretes bile.
Yeah, it makes bile. But wait, there's more.
Detoxifies chemicals.
Totally detoxifies. So it has all, in fact, when you get into pharmacology later, you're going to learn about some of the main enzyme pathways in the liver that are used to break down most of the drugs that you take. So the liver is what, it's why if we talked about drinking your tequila shots and getting buzzed, it's why you don't stay buzzed. The liver enzymes are breaking down that alcohols and then getting rid of the byproducts. So, the drug breaks down. So detoxifies things. What other things does it do?
Doesn't it release hormones?
That I don't know if it does. It's having to do with the blood. It makes the plasma proteins though. So it does release things into the blood for sure, which are the plasma proteins. It makes your plasma proteins. So it's doing a lot of really critical stuff. As opposed to the spleen or the appendix, you can't live without your liver. And it does have a limited capacity. Like, normally it's very regenerative. In general, if you do a major insult to it, it can grow back. When I was in grad school, I had Hep A and it did a number on my liver, but about a year later, my liver function was back to normal. I had a friend who donated his liver to another friend who had liver cancer. And what happened was they took half the liver out of the guy who had the good liver, and both of the livers regrew into full-size functional livers. The donated liver in the recipient became a full-size functioning liver, and the guy who donated his liver grew back to become a full-size functioning liver. The only downside was he lost part of the surgery. They couldn't preserve the gallbladder. But in terms of the basic necessary function, you can cut out half your liver and it'll just grow back. It's different if there are constant insults, like in alcoholism and things like that, where you are, constantly like kind of overwhelming its ability to detoxify, and the cells, eventually the cells start getting replaced by scar tissue and fat and stuff. And that, in which case, that's like cirrhosis of the liver and stuff like that. At that point, the liver is not going to recover, but it has a pretty amazing ability to come back.
Is that over like years of alcoholism, or can that happen within like, I don't know, like 5 to 10 years of alcoholism?
5 to 10 years sounds like a long time to me.
Well, I just feel like I know there's people who live their entire lives with alcoholism. So I was just wondering, like,
right.
for
So
sure.
that, I don't know. I don't know enough about how much you have to drink, how how much individual variability affects it. So I don't know the answer to that. What else? So liver, just, it's pretty, it does so many different things. It's kind of crazy. So any questions about any of all that stuff? Okay.
I had one question. I was just curious, going back to the
Seriously.
small intestine, of why sometimes what will happen to make it twist and then stay. And I know that can be really dangerous, or like why that's happening.
So I don't know.
Okay.
I don't know what I've heard of that, but I don't know what causes that to happen.
Thank you.
There it is. Modules. All right, so I just created this thing, cadaver dissection, digestive system. So a couple of these are from those Wisconsin ones. And then there's this Indian dude who actually has some, these kind of interesting ones. So I have a few links to some of this guy as well. Here, I'll show you this. This one, here, 520. I'm going to show you 520 on this one.
So this is a quick demonstration of the
Okay, so here he's opening up the jejunum versus the ileum. So you can really see the difference of folding. Again, the jejunum, where there's going to be a really optimization for absorption, ileum, which is not going to have as much folding.
Next, we open up the two loops of the intestine and we hold them side by side to show you the difference. The jejunum, we notice the mucosal folds are very thick, very prominent, and very large, and more dense, more numerous. These are known as plicae circularis or valvulic ornamentes. This extensive mucosal fold is required for absorption purpose. And this is what gives the feathery appearance on X-ray when we do a barium mean follow through. This is a barium mean follow through to show you the feathery appearance of the jejunal mucosa. In contrast, in the ileum, the mucosal folds are thin, few in number, and in places they are almost absent. This is what is referred to as the structureless appearance in radiology. So this is another important difference. And the final difference between the jejunum and the ileum is, that is microscopic, is when we do a section and we see in the ileum. So
he does a really nice job of really showing. That was something that we actually do look at in the cadavers in our lab. I've cut little windows into the ileum and into the jejunum, and it looks just like that. So just kind of really giving you a sense of how folded up the jejunum is to maximize absorption of the nutrients across the epithelium there. So that is nice. This one here, this is showing just the very beginning of this one, gives you a sense of the greater omentum. Like when you first open up the abdominal cavity, you basically see a big apron of fat.
Good day, everybody. I'm Sanjay Sanyal, Professor Department Chief. So we have opened up the abdomen. This is a supine cadaver. We are standing on the right side. The camera person.
So just to orient, this is the top. Right here is the bottom. This is the head side, the caudal. This is like the liver up in here.
This is also on the right side. Just to bring up to speed, this is the posterior layer of rectus sheath that we have opened up on either side, and you can see the posterior layer of rectus sheath has got the extraperitoneal fat and the parietal peritoneum here. And we have
and you also notice the parietal peritoneum. That is going to be the layer of the peritoneum that is actually against the abdominal wall. So there's the sheet of peritoneum, the visceral one that's wrapping around the organs, but then obviously the wall, the internal wall of the abdominal cavity is also got the parietal peritoneum, the lining on its wall. It's just another one of that slippery layer of simple squamous epithelium.
You can see the posterior layer of retinal sheath has got the extraperitoneal fat and the parietal peritoneum here, and we have reflected all the structures. And this is the linea alba, which also we have reflected up. You can see this yellow structure here. This is the greater omentum. These are the coils of the small intestine here. This is the liver that we can see here, and in front of us we can see the stomach here, and a little bit of the duodenum. And of course, on the side we can see a little bit of the large intestine. We are going to demonstrate the greater omentum, the stomach, principally in this particular dissection. The greater omentum, embryologically, it's a derivative of the dorsal mesogastric. When the stomach rotates, it comes in front. It hangs down like an apron in front of the abdominal organs. What are the parts of the greater omentum? We can see we have cut it out here, and we have reflected most of the greater omentum just to show the portion of the greater omentum which extends from the greater curvature of the stomach to the transverse colon.
I have a question.
Gastrocoagulum. And I'm going to mention the clinical significance of that just a little later. This greater omentum, continuation of that goes to the spleen.
All right, so just to give you a sense, there is this huge fat apron sitting in front of everything there. We'll see it again in some of these other videos as well.
Can I just ask you?
Question? What is your question?
So the peritoneum
Uh-huh.
and the mesentery. So the peritoneum is that covers the digestive tract, and the mesentery is the inside one. I want to.
No, peritoneum includes the mesenteries. The peritoneum is the whole serous membrane as a whole. Here, I'm going back to here. Let me see if I can, let me find the best picture of it. Here, I am going to share this again. Right, so if I go pointer options, right, so all this stuff, this is all mesentery. All of this, I mean, no, mesentery is a double layer. So here there's like, comes down, wraps around, then it comes back. Wherever there's this double layer is a mesentery. Comes down, wraps around. When becomes a double layer, it's called a mesentery, but it's just a double layer of the serous membrane. I don't know if that makes sense. Serous membrane wraps around, comes back. Now it's a mesentery. Comes down, wraps around, comes back. This double layered area, that's called a mesentery, but it's just another aspect of the peritoneum. So the peritoneum, the parietal peritoneum lining the wall of the abdominal cavity, the visceral peritoneum wrapping around the organs, and wherever it kind of wraps around and then doubles back on itself to create a double layer, that's called a mesentery.
Okay, thank you.
I have another question. Why is it that there's so much fat buildup in that front apron part? It
just happens to be one of the places where the body stores fat. I'm not sure. Maybe there is.
So specified.
It's also in the mesenteries around the, the primary mesenteries around the organ, the intestines here, like that really obese dude near the hallway. It's crazy how much fat is around his intestines. There's so much fat in there. It's, yeah. So yeah, I don't know why that happens to be where it sits, but that is definitely one of the places it gets deposited.
Doesn't it have to do with that there's no like bone barrier there to protect it?
It could be about trying to keep it protected. Yeah, that would be a good reason as any. Yeah, adding more padding for unprotected organs behind the abdominal wall. I don't know though. All right, let's continue back to here. So we can actually speed her up a little bit.
So we're going to open the abdomen, examine some of the structure of the peritoneal cavity, including the mesenteries, and also take a look at the inguinal region and femoral region.
Actually, I speeded her up too soon. And only the first half of this is useful for us right now. Inguinal region is down below.
We're going to open the abdomen, examine some of the structure of the peritoneal cavity, including the mesenteries, and also take a look at the inguinal region and the femoral region. Now, the first step in this process is to reflect some of the anterior abdominal wall muscles, such as rectus abdominis, that you did in a previous dissection, and then to make a large X-shaped incision through the anterior abdominal wall. And now we've removed the umbilicus in this cadaver, but make that cut of the X just above the umbilicus so that later we can watch how the umbilical vessels come to and from the umbilicus. Now, if you reflect these flaps back, you can see that there are two layers to the peritoneal sac, just like there were up in the chest for the pleural and pericardial sacs. And we've left a piece of the parietal peritoneum, a nice filmy layer here, the outer layer of the peritoneal sac. There is a layer of connective tissue called transversalis fascia that would bind this parietal peritoneum to all the inner surfaces of the abdominal and pelvic cavities. Now, if we pull that back, you can appreciate the visceral layer of the peritoneal sac that is coating all the internal organs. Now, for all these organs to receive their adequate blood supply, there are structures called mesenteries, where the peritoneum is going to form a double layer that the vessels and nerves can run through to approach all of these organs and provide them with the innervation and the blood supply that they need. Now, to get better exposure of the abdominal cavity so that you can see in here, I'm going to do something that's really not part of the dissection, but will help us expose the abdomen better. And that is to cut off some of these dangling abdominal wall muscles so that we can see down inside as we're trying to instruct you how to do the dissection here. Now that the abdomen is opened well, we can see some of the derivatives of the embryonic mesenteries. Remember from the embryology lectures that the ventral mesentery only persists in the region of the foregut, that portion of the digestive tract that continues down through about the first or second portion of the duodenum. So beyond that level, at the levels of the mid and hindguts, there will be no ventral mesentery. Now here we can see the adult derivatives of the ventral mesogastrium, or mesentery of the foregut. I'm pulling back the ventral body wall here, and there's a portion of that mesentery that continues down to the liver. It's called the falciform ligament. And there seems to be some scar tissue in this cadaver so that it's shrunken up and a little bit more tight than it is on most cadavers. But this falciform ligament would be a portion of the ventral mesentery. And the liver develops within that ventral mesentery, so we'll see another portion of it standing between the liver and the main gut tube, the stomach and the duodenum at this level. And so this derivative of the ventral mesogastrium is called the lesser omentum. And I'm sticking my finger through an opening called the epiploic foramen. So all the tissue between my finger, which you can't see, and right about in here is called the lesser omentum. Now this is all stomach, and you can't really see it visually necessarily, but the sphincter between the stomach and the duodenum is right about here. You feel a nice dense thickening of the smooth muscle there in the wall of the organ. So that this portion of the lesser omentum spanning between the liver and the stomach is the hepatogastric ligament, whereas this portion a little bit more toward the right, spanning between the liver and the duodenum, is the hepatoduodenal ligament. And that in particular is important because it's got the structures of the portal triad contained within it: the common bile duct, the hepatic artery, and the hepatic portal vein. Now, the rest of the mesenteries that we're going to see are all derivatives of the dorsal mesenteries that develop originally. And there's a very prominent specialization of the dorsal mesentery of the foregut here. It's called the greater omentum. It's attached to the greater curvature of the stomach and then balloons out and forms this apron-like structure hanging down over many of the other abdominal organs. Now let's pull up the greater omentum, and we can catch a glimpse of the transverse colon, portion of the large intestine coming across, and it's got its own dorsal mesentery. This would be the transverse mesocolon extending from the posterior body wall up to the transverse colon. And in the next dissection, what we're going to find is as we peel apart the layers of these different mesenteries, we're going to expose the blood vessels that supply them. And the last major mesentery that you can find within the cadaver is the dorsal mesentery of the small intestine, the so-called mesentery proper. And again, when we peel apart the layers of this mesentery, we'll see very elaborate blood supply and nerve supply.
Right, so this is always like, this is kind of showing how the intestines are not just a tube just stuffed in there. It's actually hanging off the back of your body wall by this double layer of serous membrane called the mesentery here. So as you kind of pull it in, here you can't really see it. Like, it's actually in the pigs, especially if they've been like injected with blue and red for their arteries and veins. It's actually beautiful. You can just see all sorts of beautiful little blood vessels running all through here. This is where all the blood vessels are running and nerves to supply and bring back blood from the intestines here. So this is not just slippery. It's also kind of supporting all of the vessels and nerves that are serving the intestines here.
Innervating all of these different organs. We're going to take you on a quick tour of the abdominal organs, just so you have seen them and are oriented and find them in your own cadaver. Of course, we've got the stomach here in the upper left quadrant, and we've already taken a look at the liver, and this would be the gallbladder tucked underneath its inferior edge. Now let's follow along the length of the GI tract, and we've got the stomach again. You can't.
I should mention the gallbladder is always like dark green, because bile is this kind of dark green. So the gallbladder, you can't ever miss it. It's this really dark green little sac on the inferior aspect of the liver. So it's really, it's really easy to see.
Can't necessarily see it, but you can palpate that pyloric sphincter that separates the stomach from the duodenum. And I can follow the duodenum a certain distance, but then it disappears where it travels right along the posterior abdominal wall and will cross to the opposite side. So to find it again, we need to pull the transverse colon out of the way. And now here on the left side, we've got the duodenum coming back into view. And as soon as we get a mesentery established, we make the transition from duodenum to jejunum. So looking at all these loops of small bowel, about half of it is jejunum, and then the second half of it would be the ileum. And we're not really concerned about looking at these grossly and making the distinction between ileum and jejunum. Just realize about half is jejunum, about half is ileum. Now, as you follow the ileum along, you're going to come to an abrupt increase in diameter where we reach the end of the small intestine and the beginning of the large intestine or colon. And where this happens, there's always a segment of the large intestine that hangs down or pooches down a little bit. That's called the cecum. And often this is where you're going to find the appendix dangling down, right at the ileocecal junction. Now, from what we've seen, there is no appendix in this cadaver, but that's where you should expect to find it if the cadaver's got one that hasn't been removed. So that would be the home of the appendix. And now we're going to see that the large intestine travels up along the right side of the body. This would be the ascending colon, and then travels across the body as the transverse colon, and then travels inferiorly or descends on the left side of the body as the descending colon. Now, in this particular cadaver, the descending colon is very small. It's probably about as big as one of my fingers in diameter, and normally it's at least a couple of inches in diameter. So there seems to be some obstruction in this cadaver. You would normally find something larger. Now notice as I've been flipping organs around here, some of the organs are really plastered against the posterior body wall. And these are organs such as the descending colon that have lost their mesenteries and have become retroperitoneal, or have assumed this position behind the parietal peritoneum. And as you go through your dissection, try to identify those organs that have lost their mesenteries and are now secondarily retroperitoneal. Following down the descending colon, the next segment that we would find is the sigmoid colon. And again, the diameter of the sigmoid colon in this individual seems to be quite reduced. We do establish a mesentery again with the sigmoid colon. And then as that organ straightens out and heads down into the pelvis, we get to the level of the rectum. For the next part of the section 6, we're going to...
All right, so that gives you a kind of a nice sense of what those mesenteries look like. And although I wish there was higher resolution. The last video, it's back to that Indian dude. It's got a nice dissection showing the whole colon kind of pulled out. So you can see the different segments more clearly and see the tinea coli and the haustra. So I want you to see that. That will be like the last one of these here.
Dr. Sanjay Sanyal, Professor Department Chair. This is a demonstration of the entire large intestine. You can see the cecum here. We can see the ascending.
Right, so this is, he's totally like cut it out. So again, here was the ileum going in. So the cecum here, which is really kind of wrinkled up, but it's that big kind of pouch-like beginning of the large intestine. Going to go up to the ascending colon, transverse colon, descending, sigmoid.
Colon. This is the hepatic flexure. This is the transverse colon. This is the region of the splenic flexure. And this is the descending colon, continuing as the sigmoid. And then it will go into the rectum. Let's take a look at certain parts of a typical segment of the colon. Straight away, we can see these longitudinal bands. It's then running around the entire length. This is known as the tinea coli. These tinea coli are condensed, thickened portions of the longitudinal smooth muscle of the intestine. Then we have the next structure, these fatty projections. These are attached to the tinea coli, and they are also present all along the length. These are known as appendices epiploicae, which can sometimes get inflamed or...
Again, we're calling them epiploic appendages.
They undergo torsion, in which case it is known as epiploic appendagitis. The next thing we notice is the sacculated appearance of the entire large intestine, the colon. The sacculations of the large intestine, these are referred to as the haustri. And the haustri are formed because the total length of the tinea coli is slightly shorter than the total length of the colon, which is 1.5 meters. And as a result of which, the whole segment of the colon has been concertinated, and that is what has produced these projections, which are known as haustri. Most of the colon does not have a mesentery. It is attached to the posterior abdominal wall, the ascending colon and the descending colon. Therefore, they are retroperitoneal. However, this segment of the colon has a mesentery, and part of which has been retained here, and this is the transverse mesocolon. Likewise, the sigmoid also has a mesentery, and that is known as the mesosigmoid. These are the two important places, and of course, the appendix also has a mesentery, which is known as the mesoappendix. Now, let's take a look at three representative areas of the colon. First, we have opened up the cecum. The cecum is the only portion of the colon which is as wide as it is long. 7.5 centimeters in length and 7.5 centimeters in width. This is the terminal ileum, which is opening into the cecum. So this is the ileocecal junction as seen from outside. We have opened up the cecum. We can see this opening here in front of us. And if I put my probe inside, it will go into the ileum. This is the ileal opening. Because this is a cadaver, this is looking like as if it's a valve with a superior lip and an inferior lip. But actually, this is not a valve. In a living specimen, this will be a small projection, and this is actually referred to as the ileal papilla. And at the tip of the papilla is the opening. So it is not a longitudinal slit, rather it should be a circular opening. So this is the ileal opening. Ideally, we should be able to see the appendicular opening, but in this case, this is the approximate location of the appendix. The appendix has been removed, and we can see a remnant of a suture material which is present here, which had been tied to the appendicular artery. So this is the location, and we can see some additions here. This is the portion where the appendix was located. This patient has undergone appendicectomy. I have to tell you how the tinea are named. The tinea coli, there are 3 of them, and they're named in relation to the transverse colon. This tinea coli that you see here, this was in relation to the great.
So, yeah. You do not need to know the specific names of the different tinea coli. But I think this does do a good job of showing you, at least to some degree, what these different sections look like, what the mesenteries look like in real life, how the intestines and the stomach and the liver and everything are kind of held in place and kind of all wrapped up by the peritoneum. So yeah, I do want you to have a basic understanding of the peritoneum and how it kind of envelops the abdominal viscera like that. What else? I spent a few moments. Let me share, new share. Let's see. Wait, what just happened? Hold on. So here is the model again. Here I just have the liver, and I'm going to turn that off. There's the liver. This is obviously a model, but it kind of more prettily shows the falciform ligament. So you have the right lobe, and there's a right lobe of the liver, the left lobe of the liver, and in between is that falciform ligament. That's that falciform ligament there, which is actually another mesentery. And then on the bottom of the falciform ligament, that's that ligamentum teres, or a round ligament. That is that little nubbin there. If you followed it, it would actually go all the way to the backside of your belly button. You know, if we add in the person here, let's add in the person. I want to fade even more. Oh, I'm not doing a good job of it. Let's turn off the skin. But basically, it's actually pretty cool. When you have the actual cadaver, you literally can follow this little dealy bopper to the person's belly button and know that, 90 years ago, this is where the guy was receiving blood from the placenta of his mom when he was in the womb. Underneath here, we can actually see the gallbladder. And then we can see these different ducts we've been talking about. And here's plunging into, I'm going to hide the, all right. I wanted to hide that. Okay, so we have basically, this is the pancreatic duct coming from the pancreas. This green one is that common bile duct, or actually just called the bile duct, coming from the liver. And here you can see we have this, oh, back. We have right here, oh my God. I keep thinking I'm getting better at this, but I'm still, there we go. All right, so now we have, here is this common hepatic duct. This is where bile is getting made by the liver and is getting delivered down. That hepatopancreatic sphincter of Oddi would be down where I'm making the mouse go around. That's heading into the duodenum. So if that is blocked so the bile can't move down this way, then it just backs up this cystic duct here and then ends up just getting into the gallbladder where it kind of gets stored and concentrated. And then under the influence of cholecystokinin, the gallbladder will contract and squeeze the bile down the bile duct, you know, down into the duodenum. So I think that's just kind of nice. I liked that you could actually kind of see that in a kind of more three-dimensional way. And also just the falciform ligament and the round ligament make more sense here. So if you see it here, then think about it again on those pictures of the cadavers. It will probably make more sense.
So the ligamentum teres and the round ligament, they are the same
Yeah.
synonym?
Yeah, they're so round ligament of the liver and ligamentum teres are the same. So they are synonyms.
Thank you.
Sure. What else do I want to say here? In terms of the microscopic stuff, you know, you definitely want to, let me go back into the modules. So under histology resources, I put this thing called more digestive slides. You know, here's some of the colon. Now that we've been talking about this, hopefully you can think of these. These are these crypts. Go and maybe get my pointer here.
Your screen is still on the complete anatomy.
What's that? Oh,
The screen that's showing it us is just the complete anatomy right now.
oh my God, thank you. Thank
Yeah.
you very much. Okay, in fact, let me go back so I can show you where this is. All right, so under where it says histology resources, so we had that virtual histology lab, which has got a lot of good stuff. I also made a little page here called more digestive slides. And if you click on that, so here is the colon, and you know, each of these are the entrance to one of these crypts. And so this is kind of the entrance to the gopher hole. This is like kind of going down the gopher hole. And all of these light colored cells are the goblet cells that are making the mucus, that are just basically making all the mucus that's making everything all slippery and keeping your kind of feces moving along. So this is a pretty classic looking colon where you have these crypts, basically these kind of gopher holes, I'm calling them, lined with goblet cells. And here again, entrance. You know, if it gets cross-sectioned this way, then they don't look like gopher holes anymore. Then they start looking like flowers, like I was saying. So let me clear this. So these pictures here, this is the same slide. is just rather than going down the gopher hole, you're looking at the gopher hole from the top. And then you've got kind of loose connective tissue of the you have the connective tissue around the epithelium. But each of these little round dealies is a crypt. This would be like, and this would be the lumen down the center of the crypt. And these light cells are the goblet cells, the petals of the flower. So again, that's just the same thing. Like this is Instagram. I remember one time I decided it would be fun to post this on Instagram. So I used an Instagram filter and it made it even prettier than it was. And so I put this in here because I'm proud of this picture. I think this could be art. It is art. This is also colon. You can tell because you've got the crypts, which are full of goblet cells. Again, we got crypts and all these goblet cells, these big light colored bubble looking cells around. But then this is showing more. This is also showing down into the submucosa really well. And muscularis externa, circular layer, longitudinal layer that you're looking at head on. And then this actually shows the myenteric plexus. These things in between the two layers of the muscularis are the myenteric plexus of Auerbach. So that's another one you can practice. You can have your crypts of Lieberkühn where you're getting like the and when you're doing your plexus of Auerbach, you can kind of go to the back of your throat and try to get that more guttural Auerbach. What else? Clear drawings.
So one at the very outer part, those are tinea coli of the muscularis externa?
Yeah, so there's kind of concentrations of longitudinal muscle. Those are the tinea coli.
But they're not called longitudinal muscle.
I mean, they're the longitudinal muscle that make they're called tinea coli because they look like ribbons from the outside. But why they're actually there is because of the longitudinal muscle being concentrated in strips.
Okay, thank you.
Yeah. Well, this is getting into, again, what organ is this?
Is that the salivary gland?
It's totally the salivary gland. And again, there's a couple of total giveaways here. And you've got the classic looking kind of mucus cells and serous demilunes. And then we also have these cuboidally constructed ducts. So we're in a gland because we're all full of ducts, and it's got these classic looking mucus and serous cells. So this is a really nice looking salivary gland, if I say so myself. Oh, and again, really nice looking. Here's a vein. It's so obvious. You can see the blood vessels with the thin wall and then the blood cells, like the little red blood cells sitting inside. Oh my God, this is an amazing one. You can just see all the cool little blood cells in there. This is showing the same thing from a lower magnification where you can just see ducts everywhere and also clusters of both mucus cells and serous cells. This is a tooth. So decalcified, so there is, they dissolve the calcium, the mineral component, so it's just like the osteoid part that's left of the dentin. This is the pulp cavity. This is the main part of the tooth. This is going into the pulp cavity at high magnification, and you can see these little dentinal tubules. I was talking about it has those little, because unlike bone, unlike compact bone where you have the osteons with the blood vessels, there's no blood vessels going through the dentin in the tooth. Instead, you have these little odontocytes that send their little processes down in there to help maintain the dentin, the matrix of the tooth. Somebody asked about whether teeth can repair themselves. I looked into that a little bit. And I think they can maintain and repair the dentin. So the dentin can actually fix, but the enamel is something that gets deposited by these special cells when the teeth are still down inside the bone, but before they're erupting. So I don't think enamel ever gets repaired. So the teeth, the actual bone part, the dentin part of the tooth can repair, but the enamel part is kind of one shot, like protective coating. That is when that kind of gets messed up, you gotta, that's it. Doobie-doo. And back to this histology slide box, gastrointestinal tract. Right, now that we've been talking about this more, this is a beautiful shot of the ileum. We know part of the reason why it's really easy to see the villi is because the ileum is not so folded. When you're in like the jejunum, there are so many freaking villi, it's like you can't see anything. It's just everything's piled on top of each other. But when you're in the ileum, it's much more gentle folding, so you can actually get nice pictures showing the little finger-like projections. So here you can see the columnar epithelium, these little enterocytes going. You can see the lamina propria, the loose connective tissue that's kind of underneath the actual epithelium. You know, you can't actually see it, but you know that the blood vessel, the capillaries and the lacteals are going up each of these fingers to absorb the nutrients and the fats. This is showing the submucosa here. Here's the submucosa is going up as part of one of the circular folds, the plicae circularis. Here's a big blood vessel, and we talked about blood vessels being in the submucosa. Here's an arteriole with its smooth muscle. It's probably a vein with much thinner walls. Then below that, we've got the muscularis externa. Here's the circular layer. Here's the longitudinal layer. So if you saw this, I would feel completely fine putting this on a test, and you should know that you are in the small intestine. You know, it's really, nothing else looks like this because it's got really obvious finger-like projections like this. Whereas if you're in the stomach, let's actually get a, let me find a good spot. You know, instead you've got like these kind of gopher holes going down and going down into regions just full of all sorts of glands. I would get you a better picture, you know, one where you can actually, I do have some better pictures in this. But again, it's really hard to get a good sideways picture because everything is so dense. It's very common to cut the tubes crossways. What about here? Yeah, like here. Here you can see like the little gopher hole going down, and then down here it's just full of glands. Here's another gopher hole, another little gastric pit going down to where all of these glands are. This looks so not like a bunch of fingers, right? So you should be able to look at this and know you are not in the small intestine. And then we also know we're not in the colon as well, because the colon would just have gopher holes with goblet cells to make mucus. They don't have this whole crazy glandular structure underneath. So if we go to the colon, let's see if you can see that, you know, the gopher hole just kind of goes down and that's it. There's not a whole lot more to it. And again, if I put one of these on a test, I would not put it in an area that is ambiguous. I would make sure I put it in an area that was very obvious. You do not need to know the appendix. Oh, here, this is good. Is this? Yeah. Again, these crypts, basically like a gopher hole lined with goblet cells. And then all of a sudden we're in the submucosa. It's not like the gastric pits where you go down into this crazy zone full of glands and stuff. So hopefully the colon makes sense to you as well. Don't worry about the rectum or the appendix. Don't worry about knowing where you are in the stomach. I will show you the jejunum just for, so you can, oh, this is actually pretty gorgeous. You rarely get something this pretty. Here you can see how much longer and more developed all the villi are compared to what we saw in the ileum. Plus you've got all these things cut across here. Let's get another picture of the jejunum as well. Right, it's almost impossible to see really pretty fingers because things just get, the fingers are going, they're really long and they're entangled and you end up getting cross cuts of the fingers everywhere. So whenever you look at the jejunum, it's always looking like a whole bunch of entangled fingers that have been cut in every direction. Whereas when you get a nice picture of the ileum, you're much more likely to like, oh, look at that, a very like solo, you know, these are like socially distanced villi. You could probably make it socially distancing as a villus in the ileum, but there's no way you could do it in the jejunum. The jejunum, everything is so dense and packed on each other that you just, yeah, whatever. So let me stop this. So are there any questions about all of that? You know, things
So
that,
our quiz is gonna stop at the colon basically, or it's on Tuesday?
yes, exactly. So up through today. So the quiz will be, yeah, up through today. And that'll, yeah, and that gives you the whole weekend. And it'll be good for studying for this anyway, because the main exam is coming up